welcome back to 15 minutes question answering. So yesterday it was a bit messed up because we misinterpreted some questions, and um, some answers were placed in a different scenario. So yesterday's video contained some mistakes. So today we are going to reassign those topics, revisit those questions once again by some careful scrutiny, and then we are going to answer them once again. First question: the isoelectric point of alanine is 6 if alanine is dissolved in a buffer of pH 3 and subjected to electrophoresis. Answer is B. It will migrate to the cathode. Okay. Then the rate of migration of a protein in an STS polyacrylamide gel is not influenced by B. Charge of the protein. Then third D, which of the following statement is false? Sodium dodecyl sulfate is a cationic detergent. That's the false statement. 3D. Sodium dodecyl sulfate is cationic detergent. Then SDS is used in polyacrylamide gel electrophoresis of a mixture of proteins for the efficient separation on the gel. SDS in this experiment is used to D. That means have uniform charge density on the proteins. Then sodium dodecyl sulfate upon addition into water can form spherical micelles. In this micelle, the hydrocarbon tail would be 5B. B means in the interior of the micelle. And if the pH becomes lower than the isoelectric point of a protein, then how will the protein respond in an isoelectric system? If the pH become lower than the isoelectric point of protein, then how will the protein respond in an isoelectric system? It will 6B. Migrate to the negative pole. Because it's lower charge than isoelectric point. And which of the following electrophoretic procedure does not depend on the charge of the protein? Does not depend. C. Polyacrylamide gel electrophoresis and sodium dodecyl sulfate doesn't depend on charge of the protein. We just mentioned earlier. So in two-dimensional electrophoresis, proteins are separated 8A. First by their charge and then by their mass. First by charge, then by mass. Then two-dimensional 2D gel electrophoresis performed under denaturing conditions can be used to separate proteins according to which of the following characters? 9. D. Isoelectric point, subunit molecular weight. And a mixture of amino acids consisting of all these types of amino acids was placed in a center of a paper strip, moistened with a buffer of pH 6 and electric current applied. The migration of amino acids was as follows. Given like that. It's 10 correct answer is actually C. So C is the correct answer. Glycine retained near the origin. Glycine remained near the origin. Lysine, arginine and histidine moved toward the cathode, while aspartic acid and glutamic acid moved towards the anode. And SDS and NaCl are used in the purification of genomic DNA from peripheral blood samples. SDS and NaCl are used in the purification of genomic DNA from peripheral blood samples. Their function and the process are respectively 11D. D means denaturation of protein, salting out of proteins. And the relative viscosity of a protein solution is affected by temperature, shape and molecular weight. Proteins may be separated according to size by 13C. Molecular exclusion chromatography. Size. The dilution of a protein from a size exclusion chromatography depends on 14B. The size and shape of the protein. Size exclusion chromatography is obvious answer. Then a solution of a mixture of proteins is loaded onto a gel filtration column that has a size exclusion limit of 75,000 Dalton. The proteins have the following molecular mass in Daltons, like given. The proteins will elute from the column in the following order. Which order? 15 B, B, D, B, C, A, D, B, C, A. That means from higher to lower. Nothing special about that. Then a mixture of lysine, aspartic acid, tyrosine, and alanine is passed through an anion exchange column at pH 8. What is the likely order of elution of amino acids when eluted with the buffer of reducing pH gradient? 16 D. Lysine, tyrosine, alanine, aspartame. Anion exchanges preferentially bind proteins with 17. Correct answer is found to be A. Acidic isoelectric points. Then separation of one protein from one other proteins on the basis of molecular size can be achieved by 18A. Polyacrylamide gel electrophoresis. And a protein was purified to homogeneity. Determination of the molecular weight by molecular exclusion chromatography yields 60 kilodalton. Chromatography in the presence of 6M urea yields 30 kilodalton species. And the chromatography is repeated in the presence of 6M urea and 10M beta mercaptoethanol, a single molecular species of 15 kilodalton results. The protein is a 19D homodimer of 15 Ka. Electrophoresis of a purified protein named X in the presence of sodium dodecyl sulfate and 2 mercaptoethanol shows a single band of 45 kilodalton. In gel filtration column chromatography, protein X elutes between alcohol dehydrogenase and beta amylase. How many identical subunits is protein X composed of? 20 D. D means 4. Then molecular weight of an unknown protein can be found out by 21A electrophoresis. A spectrum shown below is of a sample dissolved in buffer pH 7 at 1 cm path length cuvette. The sample is most likely to be 22D, a solution of protein. And although protein concentrations can be estimated by measuring the absorbance of a protein solution at 280 nm, this technique does not yield an accurate measure of absolute protein concentration. Why? 23. Correct answer is C. That means the aromatic amino acid content varies from protein to protein. And according to Beer-Lambert's law, what will be the absorbance when I is equal to I0 by 10? 24. B. B means 1. Absorbance 1. A solution of tryptophan has an absorbance at 280 nm of 0.54 in a 0.5 cm path length cuvette. Give the absorbance coefficient for tryptophan is 5.4 into 10 raised to 10 L mole inverse centimeter inverse. The concentration of the solution is found out to be 0.2. Okay. The molar absorption coefficient, extinction coefficient of NADH at 340 nm is given, whereas NAD does not absorb at 340 nm. What absorbance will you observe when light at 340 nm passes through a 1 cm cuvette containing 10 μm NADH and 10 mm NAD? 26. B. Fluorescence, 0.062. Change in protein conformation can be detected by circular dichorism. Conformation change can be detected by circular dichorism. Using circular dichorism, one can obtain 28. Correct answer A. Relative proportions of alpha helixes, beta sheets, and loops in proteins. Yeah, that's right. The secondary structure of a protein cannot be determined by one of the following techniques. Cannot be determined. That means all other techniques are capable of determining that. So 29C, fluorescence spectroscopy. But NMR spectroscopy, X-ray crystallography and circular dichroism can be used to measure the secondary structure of a protein. To determine that, what is the most common approach for the determination of precise 3D structure of globular proteins? What is the most common approach for the determination of precise 3D structure of globular proteins? 30. Correct answer is D. X-ray diffraction. Then the void volume of a gel filtration column. A monomeric protein with a non-molecular weight of 25 kilodalton elutes at a volume of 45 ml. The protein that you are trying to purify elutes at a volume of 35 ml. Which of the following is a valid conclusion? Which one is the following? Which one of the following? 31. Correct answer is C. That means your protein is like this. So, M greater than 25 kilodalton. MR. Monomeric protein, I guess. Molecular weight. Then a homogeneous protein of native molecular weight give a single band of molecular weight. That's also given on STS page in presence of beta-mercapto-ethanol. In terminal analysis gave two amino acids alanine and leucine in equal proportions. Hence, 32C. Protein has two polypeptides linked by disulfide bridges. Then a mixture of four proteins are loaded on DEAE anion exchange column equilibrated with low ionic strength buffer of pH 8. Which of the following proteins would be expected to be retained on the column? 33C. C in the sense proteins with plus 7, 5 and 3. Then protein sequencing, choose the mismatch. Phenyl isothiocyanate, disulfide, protein sequencing. Next section. 1A is a mismatch. All others are found to be correct. Dicyl chloride identification of the amino terminal residue of a peptide. Chromatrypsin, hydrolysis of peptide bonds on the carboxyl side of aromatic residues. Then CNBR, cleavage of peptide bonds on the carboxy side of methionines. Which one of the following statements are correct about Edman reaction? The peptide does not have to be hydrolyzed for recovery of the reacted N-terminal amino acid. The peptide does not have to be hydrolyzed for recovery of the reacted N-terminal amino acid. Which one is correct? Is about Edman reaction. 2. Correct answer C. That means C in this end P and R. P and R. P. The peptide does not have to be hydrolyzed for recovery of reacted N-terminal amino acid. And R. R means the reaction can be carried out repeatedly on the same sample of the peptide for sequencing. Then a protein treated with fluorodinitrobenzene FDNB and subjected to acid hydrolysis yielded two different amino acids with their alpha amino groups linked to DNA. A reasonable explanation for this result is that 3B. A protein contained more than one N-terminal amino acid. The N-terminal amino acids are usually determined by Sanker's method using 4. Correct answer B. B means fluoro-2,4-dinitrobenzene dicyl chloride. Then a polypeptide reduced has yield two peptide fragments in the following sequence. Correct answer. Indicate the positions of disulfide bridges in the polypeptide. 5. 5A is the correct answer. Then chymotrypsin in the small intestine hydrolyzes peptide linkage containing phenylalanine. next questions we will be dealing with the help of next video. Thank you for listening.